Next up is metagenomic data analysis to unravel the microbial community. To explore the limitless potential of metagenomic data analysis through our comprehensive training program that is designed for beginners, intermediates, and also advanced level practitioners. So whether you are starting your journey or seeking to expand your expertise, this program equips you with the skills and the knowledge to excel in this dynamic field. Now let's get back to understand where did the metagenomics field come up from. So in the 1800s, the link between disease and the microorganisms was established, shedding light on isolated bacteria functions. And today we understand that the human microbiome consists of structured communities forming biofilms with complex interactions aiding ecological stability. And advances in metagenomics enables the study of the vast majority of uncultivable microorganisms within their natural environments. So the course covers bioinformatics analysis of metagenomic sequencing data, which includes genomic sequencing technologies, data utilization, analysis tool, as well as real-world applications. So since the 1900s, microbiome studies have made significant progress, and notably through initiatives like the Human Microbiome Project that enables metagenomic sequencing advances, allows categorization of microbiota in various human organs, presenting opportunities as well as challenges in identifying their roles in organ health and disease as well. So in the program, we will learn about these different types of metagenome sequencing techniques through different types of data that is generated. We'll majorly focus on 16S metagenomic sequencing and the data generated from it, such as OTU sequences that is annotated by mapping onto a reference database, such as the Ribosomal Database Project RDP, Green Genes, or the Silva database. And such databases contain taxonomic information for the domains of bacteria, archaea, and eukarya as well, that is based primarily on phylogenies for small subunit rRNAs, which include 16S prokaryotes and 18S for eukarya. Now, based on the mapping of specific regions amplified from the 16S RNA gene in the small subunit of the bacterial ribosome, OTUs can be assigned to belong to a taxonomic class of bacteria, yielding a proportionate view of the community. And each community can then be studied by comparing with others or between similar communities in different conditions or individuals. And such analysis includes an in-depth understanding of data preparation, reference databases, and analytical approaches to answer specific questions based on solid evidence. Importantly, sequencing data that represents only one type of microbial omics, with many more becoming increasingly available. So there are many questions about the role of microbiome members and the communities that are now being answered using next-generation sequencing genomic data, as well as mass spec approaches for proteomics and metabolomics. Indeed, the next phase of microbiome research lies in the ability to integrate multi-omics datasets that combine NGS, mass spec, and structural data with phenotypic information about the overall changes in the community and their environment. And in the program, we will go through these three different pipelines that I just briefly touch upon. The first one is the taxonomy classification. Then we have the data-to pipeline that denoises and corrects errors in amplicon sequence data, providing high-resolution, error-free sequence variants as output. And next, we will also learn about KIME-to pipeline, which analyzes and processes microbial community data, offering diverse outputs like taxonomic assignments, diversity matrix, and visualizations. And behind many of these methods are scripts that are written in popular coding languages such as R and Python. And these are also explained in the tutorials that you will have an access to as a part of the program that I'll just take you through briefly. It includes beginner tutorials on data importing, preparation for analysis, and also standard visualization of the processed data, and more specialized packages with publication-ready outputs that will also be covered in more advanced lessons in the metagenomics coursework and the case studies that we'll take you through. We will also learn about the basics of functional metagenomics that explores the genetic potential and metabolic activities of microbial communities in a given environment. So what kind of projects can be studied using metagenomic sequencing or metagenomic data? In the program, we'll go through these number of applications that includes skin conditions, food sensitivity of the gut, autoimmune disorders, cancer, antibiotic resistance, and also we'll try to understand the microbiome that is involved in drug resistance and even in ecological studies. These are just few applications that we'll touch upon that I just mentioned, but there are more such case studies that we'll cover in the program. In this program, we'll also learn to find and analyze data from such repositories as the Human Microbiome Project, which includes a data set that is called the American Gut. This citizen science project has a lot to offer for those interested in basic microbial composition of different types of samples from healthy and mildly sick individuals.